Hello, I am Shin. I am very happy to present my studies on archaeoparasitology in India and other Asian countries. This lecture is for the monsoon lecture, and this presentation will be prepared by myself and Dr. Jong, who is working for Kyunghee University as a research professor. We will summarize and present the current trends of archaeoparasitology and the future prospect of this field to archaeologists and anthropologists. Yes, and first of all, I will introduce myself. I am Dong Shin, professor of Seoul National University. I am the National Geographic Explorer and was the Cavalry Frontiers of Science Fellow of the National Academy of Science. I will summarize about the general aspects of the archaeoparasitology in this lecture. And in the second half of this presentation, so will be presented by Dr. Jong. He is a research professor of Kyunghee University, South Korea, and his topic is about the genetic analysis of ancient parasites. So he was studying on the archaeoparasitology. So his PhD thesis was a paleoparasitological study on nematode and trematode DNA. These are the parasites, ancient parasites, I mean, and extracted from specimens of Joseon Dynasty mummies, ancient samples. And he was the instructor of archaeoparasitology workshop at North American Paleopathology Association meeting. In this slide, I will talk about the paleopathology. Paleopathology means that study of diseases in history. Pathology is the studies of diseases. So paleopathology means that studies of diseases in history. We think that different diseases occurred at each stage of human history. This is for the hunter-gatherers' life. Hunter-gatherers have their own diseases. And the farmers also have their own set of the diseases. So we hope to know the changes in health and disease status of people in history. This study is paleopathology. Now I will talk about the examples of paleopathological studies in my lab. The first case is about the human tuberculosis. In this Joseon Dynasty mummies, you can see that in these radiological images, you can see that the calcifications in pulmonary hilar region. And these calcifications could be confirmed during autopsy. In autopsy, you can find the calcifications here and here. So this is the rare case of the tuberculosis confirmed during anthropological studies of Joseon Dynasty individuals. And the next case is about the rheumatoid arthritis. The rheumatoid arthritis case has the fusion of the carpal and the finger bones. Carpal bones here, and the finger bone here, here, and here. So this is a very typical signs of the rheumatoid arthritis. And this is about osteomyelitis. The arrows and asterisks indicate that involucrum and cloaca, very possible sign of the osteomyelitis in ancient bone. And this is the case of the fracture. Yes, you can see that the fracture and the malunion case of the forearm bones. This is the gunshot wound in 17th century. So it was made on the skull by the this was made by the musket. And this is the tumor case, benign tumor case. So it was made on the temporal bone of the skull. And this is the diffuse idiopathic skeletal hyperostosis. It means this disease is commonly found in the vertebral bones, like this. So in figure A and B, the asterisks indicate that the diffuse idiopathic skeletal hyperostosis. And this is the case of the cherubism. Cherubism is a very rare disorder that causes prominence in the lower portion in the face. So you can see that the figure A, B, C, D, and E. So the fat indicated by arrow and marked as W. So this part is very prominent. So comparing with the normal individual. So this was the genetic disorder, and the characteristics is the prominence of the lower portion of in the face. So in this 17th century Korean mummy, so we can find the cherubism case and reported to academia. And this is the congenital diaphragmatic hernia. This is the congenital malformation of the diaphragm, and the malformation of the diaphragm always allows the abdominal organs to push into the chest cavity, so hindering proper lung function. So in figure C and D and E, you found that the LV means that the liver. So liver could not be present in the thoracic cavity. However, you can find the liver in the right thoracic cavity. This means that the liver that must be present in abdominal cavity is moving into the thoracic cavity through the defect in the diaphragm. This is also the congenital disorder, and we can find and report this case. And this is the atherosclerosis case, very famous. This is commonly occurring in modern people, and we also found that this case even in the pre-modern Korean individual case. And this is the coronary artery disease. In the lower figure of the right column, so you can see that asterisk. Asterisk is atheroma in the coronary artery. So this makes the blood flow difficult to flow inside the coronary artery. So makes many troubles in the patient. So this, ancient individual might have been suffered from the coronary artery disease. Yes. So in 2015, I have a chance to present my works in the Cavalry Frontiers of Science Symposium. And so I summarized my studies so far and presented my works in the symposium. And the lecture could be available in this address. So you type this address in the internet, and you can find my lecture in the Cavalry Frontiers of Science site. Please enjoy. So I will explain about the parasite. So I just have explained the paleopathology, and archaeoparasitology is one of the research fields of the paleopathology. This belongs to the study of the ancient diseases. However, the study on ancient parasites have many properties of unique pattern, and there are many great achievements in this field. So I will introduce those pioneering studies to you in this presentation. So first of all, I must explain the parasite. Parasite, as you know, parasite is some very small worms impacting human by various routes. So you might know the Ascaris, malaria, and the Paragonimus. All these kind of the diseases is caused by the parasites. And the relationship between human cultural development and the evolution of infectious disease. This relationship is closely correlated. So as an anthropologist and archaeologist, you must have interested in the changes of the parasite infection pattern in history. So this is very important study to understand the human life throughout history. And now I will talk about the archaeoparasitology. What is the archaeoparasitology? Archaeoparasitology is the parasitological studies in history. And it is an interdisciplinary dialogue between archaeology, history, medicine, and even molecular biology, etc. You can see these gentlemen. They are the Dr. Araujo and Dr. Reinhardt, and they are the founder of the archaeoparasitology. They are very respectful scholar and archaeoparasitologists in the world have deeply influenced by their achievements. And to perform archaeoparasitological studies, where can we find the ancient parasite eggs? The specimens is originated from coprolites, latrine, or other waste material dumping site, and sometimes from soils of geological strata and from mummies. Now I will explain the sampling procedure used in archaeoparasitology one by one. The specimens from different archaeological sites were used for archaeoparasitological study. This is the typical archaeological site. When you hope to get the samples from the graves, you must wear the protection clothing, gowns, and head cap, and mask, and disposable gloves, and try to collect the specimens from the hip bone, and another sample must be also collected from the outside of the graves as the negative control. Sometimes we collect the specimens from the geological strata like this. And in the samples, we can find the ancient parasite eggs as seen in the figure below. So from these specimens, very well preserved ancient parasite eggs could be observed. You can see in the figures, you can see the ancient and modern parasite eggs, and even in case of the ancient eggs, the preservation state is very good. So very little difference could be observed between the ancient and the modern eggs. So now I will explain the examples of the archaeoparasitological sampling. This slide is for one sampling site of Seoul City. Seoul City is a very big city, and in the you can see that the photo of old Seoul City in 1890s, and you find that the red arrow and the site indicated by red arrow where we have sampled the soil samples from those places, and in the upper figure, so this is for now, and you can see that the same red arrow could be seen, and those two red arrows indicate the same place. And when the construction was made in the place, so we went to the place, and in that time, the archaeologists already excavated the site, and we can see that the geological strata, like this figure. So we have done the archaeoparasitological sampling from each geological strata, like this one. So you can see that the yellow and the red circles, all those circles, we could get the soil samples from the geological strata. And we found many parasite tags, ancient parasite tags like this. Another sampling site I will introduce now. This is for the site of the at the capital site of the ancient Korean ancient kingdom. And in this site, this site is 1,600-year-old one. And in this site, we have done sampling at the different places. You can see that the A, B, C, D, E, F, all these places we have got the sampling from the geological strata, and we found different kinds of the parasite tags in the specimens. And we did also sampling at the ancient potteries. This is the site of the ancient potteries, and we have sampled in from the geological strata of the reservoir, ancient reservoir of the potteries, and we found many parasite eggs in those specimens too. This is for the ancient parasite eggs found at latrines. So the site seen in the left column, those two sites are for the latrines, and we could find many parasite eggs in the specimens from the latrines. Based on the study, so archaeologists in South Korea could reconstruct the structure of the ancient latrine, ancient toilet, like this one. And now I will explain about the mummies. Mummies is very good source for the sampling of the archaeoparasitology because the preservation status is very good. So this slide explains the interdisciplinary collaboration for the mummies. And during the investigation, we sometimes found the coprolites inside the mummies. In the right figure, the materials indicated by the black arrow, this is the coprolites. This is this figure, these photos taken by the endoscopy. And in the radiology, we sometimes found fishes still filled in the intestinal lumen. So materials indicated by the asterisks are the feces, mummy feces. So during the autopsy, we collected the specimens for the archaeoparasitological studies. So in the lower row, the leftmost figure is the coprolite from the intestine, and by the examination of this specimen, the coprolite specimen, we found ancient parasite eggs. Now I will explain some archaeoparasitological cases one by one. In this case, so coffin of the Joseon Dynasty, so several hundred-year old one, was found in an archaeological site of South Korea. The preservation status was very good, as you can see here. And this was the mummy bundle. So this means that the mummy was wrapped by the clothing. So the preservation status was perfect. And the historians confirmed that she was living in the 17th century. You can see that the yellow-colored wife, the mummy was the wife, living in the 17th century. And the samples were collected from the mummy. And in the specimens, we found ancient Paragonimus eggs. Paragonimus is the river long fluke living in the human liver and make many kinds of the complications. And the ancient Paragonimus eggs were found in different kinds of the mummy organs. In case that the Paragonimus eggs being discovered in the mummy organs other than lung, we call it ectopic paragonimayasis. This means that the Paragonimus infection in the organs other than lung. I will talk about another case of the paragonimayasis. In this case, we found those parasite eggs in another mummy. You can see here in the radiological image, in the liver, we found a round mass. The round mass is indicated by yellow arrow. And when we confirmed the mass, examined the mass by autopsy, we found many Paragonimus eggs were discovered in the mass. This means that, like the other case previously described, the Paragonimus infection was found in the other organ, in this case liver, other than lung. This is also the ectopic paragonimayasis. Now I will talk about another archaeoparasitological case. This case is about the Gymnopaloides seoi. G. seoi was first reported in 1993 from a Korean woman suffering from acute pancreatitis and gastrointestinal troubles. However, in the feces of the 17th century mummies of South Korea, we found the presence of G. seoi eggs too. This means that they are the first reports of the discovery of G. seoi infection in archaeological samples. Now you can see that the Korean peninsula, the South Korea map here, and the blue dots represent the areas where mummies were found with ancient G. seoi infection, and red shadow area means that the modern endemic area of GCOE. So we know that the places of ancient GCOE are not included in the current endemic area of GCOE. This means that several hundred years ago, GCOE infection must have occurred in the coastal areas of Korea more often than now, and GCOE endemic areas must have contracted to more restricted regions throughout history. So this is the summary of the archaeoparasitological study in South Korea. So by my lab, we have examined the, in this case for the Korean mummies. So we investigated 24 Korean mummies, and we got the data from those mummies. When we examined the coprolites from the mummies, we found a different kind of the parasite eggs like this. Many kinds of the different kinds of the parasite eggs were found in different Korean mummies, and so based on the data of mummies in South Korea, so we can estimate the change in parasite infection prevalence. So this is the data based on our archaeoparasitological studies, and these are the parasite infection prevalence based on the archaeoparasitological analysis, and all these ones are obtained from the national survey on Korean population about the parasite infection prevalence in 1971, 1981, 1992, 2004, 2012, and all these data could be compared to the Joseon data. So we can see that the change in parasite infection prevalence among Korean population in history. So the infection prevalence of Korean people in history could be estimated by the results of archaeoparasitological study of ours. So the archaeoparasitological studies described above could be summarized in this book. So this book is the Handbook of Mummy Studies, and edited by myself and Dr. Rafaela Mianucci, and the Archaeoparasitology of Korea and other countries. Those information is summarized in this book. So this book will be published next year, and so I recommend this book to you if you hope to know the general aspect of the archaeoparasitology. Now I will talk about the archaeoparasitology in India. So I have collaborated with Dr. Basant Shinde of Deccan College to study on the Harappan civilization for the past several years. And we did excavation for the Rakhigali site and got many data from the site. And archaeoparasitological studies also included in the studies on the Rakhigali site. I and my team also joined the excavation. And you know, Dr. Shinde is a very good friend and collaborator in India, and we did our best to know the life of the Harappan civilization people by archaeological and anthropological researches. During the studies, when the skeletons were exposed to us, then we did archaeoparasitological sampling like this, and from the hip bone, and so with the negative control, both samples were subject to the parasitological examinations. Yes, these are the samples from the Archaeoparasitology site, and we did some rehydration, and some microscopic examinations has been done in India and in South Korea. And besides Archaeoparasitology site, we also did the archaeoparasitological studies on different sites of the India, Kota Badri, and Jilund, Balafar, and Dolabira, and Mitathar, and Karsola, and Parmana. The samples of all these sites were also subject to the archaeoparasitological examinations, but we could not get any positive data from all these specimens. So as you can see here, so our most of our studies have concentrated on the sites of this area. Those are western part of the Indian subcontinent. However, so in the modern parasitology, I mean the parasitological studies on modern Indian people, so parasitologists found that the high infection rate could be observed in these areas. All these areas showed the very high prevalence of parasite infection prevalence. However, in case of this area, the infection rate is very, very low. So we think that in this area, so ancient samples could not make the positive result like our data. And the future archaeoparasitological studies in India must be done on these areas. If we could do studies on these areas, I mean the studies on the archaeological sites of these areas might will make a significant result of the archaeoparasitological studies. Our study could be summarized in this paper published in Ancient Asia. Please see and please read this article if you have interest in our research.
Hello, my name is Jong Hoon from Gyeonggi University in South Korea. Today, I prepared a monsoon lecture for you. The title is Ancient DNA Analysis of Multiple Parasite Species Eggs Discovered in Coprolites of Mummies. In this lecture, I will tell you about the workflow from collect ancient samples from archaeological site to phylogenetic analysis. Before start, I think I must say one thing. As you know, English is not my mother tongue, so my pronounce is not much good. So I made my slides with text. If you have uncomfortable with my pronunciation, please see the text or send email to me. My email address is included in the final slide. Okay. To reveal parasitic infection patterns among ancient people, archaeoparasitology applies a variety of scientific techniques to archaeological samples. Over the years, scholars found various species of parasite in the feces or precipitates of ancient specimens by microscopic examination. Interestingly, genetic data of several parasite species could also be obtained. Large databases of ancient parasite DNA could be constructed by efforts of scholars around the world. In this lecture, first, I will show how ancient DNA analysis could be done on the parasite specimens from archaeological site, I mean the workflow. And second, the examples of archaeoparasitological study on parasite evolution throughout history could be shown in this lecture. As Professor Shin already said this, so I will mention this section shortly. In our workflow for archaeoparasitological examinations, we collect samples of ancient DNA analysis from archaeological excavation sites. The left picture shows how can we get the samples from soil strata. The soil sample was collected by conical tubes at each layer of 2,300-year-old shell mound. These are conical tubes inside the layer, as you can see in the left side picture. And this picture shows the situation about collecting samples from human bones inside the barrier, as you already seen in Professor Shin's slide. If the buried person of the grave was infected with parasites, the eggs remained in the soil precipitates upon the hip bone. And while we collect the samples from the hip bone and sacrum, negative samples must be also collected as negative control. And this is mummies. The mummies found at the excavation site are also used for the archaeoparasitological study. And to prevent contamination, every researcher wear a sterilized mask, headcap, gown, and gloves from the sample collecting part to end of experiment. And please see the left side picture. The mummies found in Korea are usually buried with a lot of cloth. The clothes worn by mummies are also worthy of archaeological research, so removing mummy cloth is done by people who specialize in ancient cloth. In this case, after removing mummy cloth was done, we finally found a male mummy who lived and died in 17th century. Because this mummy was found in the region of Cheongdo in South Korea, we labeled this mummy as Cheongdo mummy. To give an example of a workflow examination, we will follow this case in this lecture. In this case, I mean the Cheongdo mummy, at first we sampled the feces remaining in the intestine of this mummy to find the parasite eggs by microscopic observation. Before microscopic observation, samples were rehydrated in a week, like this picture. And after rehydration was done, we observed the sample with the microscope. We also checked a negative control to ensure that the sample is really contain ancient parasite eggs. During microscopic observation, we calculated eggs per gram of parasite eggs in the sample. That is a great way to check this person is really infected with parasites. And through microscopic observation, we found Metagonimus westermani eggs from mummy fishes and used this sample for DNA analysis. We first extract ancient DNA from the fishes, and then we used cloning and sequencing method for amplification of ancient DNA by polymerase chain reaction, we called PCR. The picture on the left shows how to prepare PCR. For ancient DNA analysis, every course of the experiment is done in the clean bench in the clean room. And for the DNA result more authentic, PCR preparation and polymerase chain reaction should be done in the separate place. And the right picture, the picture on the right side is a scene in which PCR is in progress. After the cloning and sequencing was done, now we got the DNA sequence from the ancient parasite eggs. Unlike the modern sample, ancient DNA is damaged and fragmented. So we amplified each small DNA fragment by PCR and used them to obtain the entire sequence. So I made PCR primers for each amplified DNA fragment length was under 190 base pair. And during experiment, we also used uracil DNA glycosylase to control carryover contamination in polymerase chain reactions. The left picture shows that the DNA amplification was successfully done, and the right picture shows the electroparagram is a plot of DNA sequencing result. And this is a large database. We call this GenBank. GenBank is genetic sequence database, an annotated collection of all publicly available DNA sequences. It is a part of the International Nucleotide Sequence Database collaboration, which comprises the DNA Databank of Japan, the European Nucleotide Archive, and GenBank at the National Center of Biotechnology Information. These three organizations exchange data on a daily basis. And DNA sequence of various species registered by many researchers have been databased in GenBank, and the number is still increasing. This picture is the result of the GenBank searching by keyword Ascaris lumbricoides. You can see more than 30,000 Ascaris lumbricoides sequences are registered in GenBank. This program is basic local alignment search tool. We called this BLAST. BLAST finds regions of similarity between DNA sequences. The program compares nucleotide sequences to GenBank database. And BLAST calculates the statistical significance of the similarity between sequences. We input DNA sequences that we found through this experiment. And we got this. This picture is result page of the BLAST search. By inputting the specific sequence into BLAST searching, similar sequences could be obtained in the closest order from GenBank database. At the BLAST result page, query cover means a percentage of query covered by alignment to the database sequence, and ident means the highest percent identity of all query subject alignment. In this result, the most similar sequence is Paragonimus westermani, reported from Shanghai, China. The query cover is 100% and the identification rate is 100%. It means this ancient DNA result of Paragonimus westermani, what we obtained from Chengdu mummy feces, is confirmed the microscopic result. If so, what can we do next? We extracted the DNA from the old parasite eggs from mummy feces and obtained the sequence. Someone will ask you, for what? What can you do with this? Okay, of course, it is also very important that we can collect samples from archaeological sites, find parasites through a microscopic examination, extract DNA, identify the sequence successfully, and cross-check parasite species. But, as you have seen before, we can use huge data sets freely available. It will give us a more fruitful result. So, I want to add one very simple analysis to get some more interesting results. Before that, this is the final result of sequence alignment based on blast searching. By this result, we can increase our knowledge about questions. So, how much difference between 400-year-old and modern Paragonimus sequences? Or is there a difference in DNA sequence between their geographical regions? Or can we distinguish Paragonimus westermani, ancient one, and other subspecies through DNA sequences? So, finally, we draw a phylogenetic tree. In phylogenetic tree, each sample is grouped according to their genetic similarity. Distance scale means how much genetic relationship each sample has, and the group is represented by node, and the group at which the splits is represented by branch. This is the branch. And this number nearby each node, we called this bootstrap value. Bootstrap value is a method for estimating reliability of phylogenetic trees. So, high bootstrap value means high reliability. A bootstrap value start 0 to 100. And the example of archaeoparasitological study on parasite evolution throughout history could be shown in this presentation. And finally, we drew the phylogenetic tree using sequences of 5 different parasite species obtained from 16th to 18th century Korean mummies. In the case of Ascaris, we performed DNA analysis for a total of 4 genes. Many people wondered if Ascaris lumbricoides could be distinguished from Ascaris sum. In this analysis, Ascaris lumbricoides was not clearly distinguished from Ascaris sum. The red dot is Ascaris lumbricoides. The blue dot is Ascaris sum. You can see Ascaris lumbricoides and Ascaris sum could not distinguish it by the phylogenetic analysis. Few differences in Ascaris gene by geographical regions, and some differences between the DNA sequence obtained from the ancient samples and the modern sample. These are ancient samples, I mean the red dots, and blue dots are modern samples. This is the phylogenetic tree about Trichuris trichiura. As you can see, the Trichuris trichiura was distinctly different from the other species. But genetic differences between ancient and modern samples were hard to find in general, but found in some cases. And the red dots are ancient samples, and blue dots are modern samples. This is the phylogenetic analysis result of Paragonymous westermani. Every Paragonymous westermani taxon was clearly distinct from the other species of genus Paragonimus. And Paragonymous westermani sequences were clustered into several groups. In case of CO1 region, Group 1 included Paragonymous westermani sequences of East Asia, while Group 2 was South and Southeast Asia, formed another cluster. We note that some sequences reported from India and Sri Lanka were also clustered into the third separate group. Among them, Paragonymous westermani CO1 sequences of Korean mummies in this study evidently belong to the East Asia group. Our result was similar to phylogenetic analysis of previous Paragonymous westermani CO1 region was clustered into the groups of East, Southeast, and South Asia. However, Paragonymous siamensis was not clearly distinguished from other known Paragonymous westermani species. Like CO1 region, Paragonymous westermani ITS2 sequences reported from East Asia, Southeast Asia, South Asia were also separately clustered in phylogenetic analysis. Our Paragonymous westermani ITS2 sequences belong to the East Asia groups as well. Taken together, our phylogenetic tree of ITS2 region was generally similar to the previous report. However, some of Paragonymous westermani ITS2 sequences of Southeast Asia and South Asia were clustered together with the East Asia group. In this study, similar finding was rarely reported exception for the research of Duan. This is a result about Clonorchis sinensis. In case of CO1, ancient sequences belong to the clade not only the Clonorchis sinensis but also with other species. As you can see in the left side picture. However, in cases of ITS1, N82, and N85, Clonorchis sinensis and other trematode species were separately clustered into different clades. We confirmed the usefulness of ITS1, N82, and N85 regions for differential diagnosis of Clonorchis sinensis from other trematode species. And finally, this is a result about Metagonimus yokogawai. Metagonimus ancient DNA sequences of Korean mummies were clustered together with the other species of genus Metagonimus in GenBank. In case of CO1 region, however, Metagonimus yokogawai formed a separate cluster that was evidently distinct from the other parasite species, including Metagonimus takahashi and Metagonimus miyatai. Okay, and this is a result of GenBank searching by the keyword of ancient Ascaris. Recently, the information of assorted types of ancient parasite is increasing significantly around the world. The right picture shows ancient Ascaris lumbricoides sequence is now registered around 90, and it will be increased more and more as the time goes. Okay, the final conclusion. As the large database of genetic information continues to grow, it will be helpful for us that information about evolutions of each species of parasite can be acquired. And genetic differences rooted in regional distinctions can be unveiled. And last, diagnosis from microscopic observation becomes valid genetically. Thank you for the attention. If you have any questions, please let me know by email. Thank you.